Hello, class. Today we're working on lab number 3, microscopes and microbial life, microorganisms. So what's a microbe or microorganisms? They're tiny, okay? Now obviously they don't all live in the marine environment, but the ones we study live in the marine environment. You can only see them or see details of them with a microscope. So they're little. Bacteria, some fungi, algae, animals, plankton, along with viruses that freeload on the cellular life. Now, viruses are not considered alive by biologists, but we included them here because they are the most abundant in the sea. There's more than 1 billion microorganisms in each liter of seawater, so they dominate the oceans. 98% of the biomass are microorganisms in this world, so we are outnumbered. We're outnumbered. The first thing, the microscope, you're going to have to name the parts. Parts A are the objectives. There's 3 or 4 usually, the 4x, the 10x, the 40x, and then the oil immersion. We don't use the oil immersion ever in class, but the 4x multiplies 4 times 10, 40 times, 10x, 10 times 10, 100 times, and then 40x, 40 times 10, 400 times, the objectives. B, stage clips come in a variety. They hold some, you have little knobs where you can move your slide around. C is the stage itself where you rest the slide. D, your light source. When I was a kid, my parents bought me a microscope when I was in kindergarten. Probably why I turned out to be such a nerd. But it had a mirror as a light source, not a plug-in, so you would have to use a lit room and focus it with a mirror. E is the base. F is the ocular eyepiece. Some are binocular. This is just an ocular one. That multiplies it by 10. That's why when I was talking about the objectives, I multiplied them by 10. So total magnification is objective times ocular. The arm, G. H is the iris diaphragm. It opens and closes. Whoop. The light source to make it brighter or dimmer. The coarse adjustment knob is I, and the fine tuner is J. Now, obviously, we're not going to use a microscope in the online portion of the class. If you were taking it live, we would use microscopes. That's one of my, now I love the online program. I think it's cool. It allows working adults to get a field class. I mean, how awesome is that? It allows us to avoid infection during times of pandemic. And it also allows us to, in state of emergencies like hurricanes, to keep going. Do work on the weekends. So it's great for our field experience, but there are some drawbacks in the fact that I don't get to fool around on microscopes all day. That's one of the drawbacks. Now, viruses we mentioned are not considered alive, even though Curtis Stuttle thinks they're alive because he says it's the most abundant life form. But so viruses are not made of cells. So then they don't carry out all the life activities that we talked about in the lecture. But they are the most abundant. Bacteria now, remember, no nucleus, simple, so small. Our microscopes, they're just going to be little pinpoints. You're not going to see much detail because they're too little. We're going to study most important bacteria that we study in this class are the blue-green bacteria, cyano, blue-green algae they used to be called. They are responsible for nearly 1/4 of the photosynthesis. That means oxygen, so there are our lungs. They fix nitrogen, which just means taking nitrogen that's not usable and converting it into usable forms. So, like a legume fixes nitrogen because it has blue-green bacteria living in its nodules. So oscillatory and anabina, you'll see when you do the lab write-up. I have pictures of them on the PowerPoint for you to sketch. I also have pictures of the letter E, upside down and backwards, showing you, and depth of field, when it comes to the microscope. So when you do your sketches, you have the some images that help you orient yourself with a microscope. Everything's upside down and backwards. And then we have some bacteria. Yogurt was there. Yogurt's a bacteria. It's a rod-shaped bacteria. Bacteria sometimes is classified by shape, as it says. That would be round, rod, and spiral. Reaction to stain, gram-negative, gram-positive. But we usually go trophic level in this class and look at photosynthetic bacteria as the most important. Protists are nucleus, mostly single cell, colonial. Algae from the tiniest microalgae to the largest kelp are considered plant-like protists. Fungi-like protists, lichens, some slime molds, but mainly, and then animal-like protists, the protozoa. So they're all eukaryotic, unicellular. Again, most common phytoplankton, we're going to look at diatoms are the most common, diatoms. Dinoflagellates for the red tide, they also sometimes glow in the dark. Our local species of red tide producing dinoflagellate is called Carina brevis. That's our local red tide. The animal-like protists, most common Globigerina, which is a foraminifera. Foraminifera are like amoebas with calcium carbonate shells, and foramen means hole. They have a lot of holes. The radiolarians are also there, and we have microscope images of them as well. Then we have some microanimals. The pteropods are crustacean. No, sorry. Pteropods are gastropods. Mollusks, gastropod mollusks, pteropods. The crustaceans are copepods and ostracods. Those are the arthropods. I apologize for the misstep. Gastropods, pteropods, those are mollusks. Crustaceans, arthropods, copepod, and ostracod. Now, lab covers as well. Complete the tables. You see comp there. That's complete a table. That's the microscope part table. And complete the magnification table, ocular times objective. Then draw the organisms on the PowerPoints. They are representative of each group. So sketch them. And being we're not in lab, you kind of get out of the preserved specimens. So you lucked out on that. So this week's this week's report is a couple of tables, sketches. Make sure you label the sketch correctly with its correct name.